All right. So, as promised, I'm going to finish our discussion about gradient sensing, the precision of gradient sensing, and then turn our attention toward multicellular problems where we can ask whether cell-cell communication can improve upon the limits that we've derived. So, we were discussing the strategy of a cell sitting in the presence of a linear concentration profile and attempting to detect the difference or the direction of the gradient by counting molecules over its spherical surface. And we proposed that a statistic that it could measure, which we called the anisotropy, would just be the sum of the cosine of the angle of incidence of each of the molecules that it counts. We calculated that the mean of this quantity is indeed proportional to the gradient. It was 4 pi a squared times g dt, which justifies the idea that the thing we care about, the relative error in the estimate of the gradient, should be simply the relative error in this statistic. And the last thing we did was we calculated the variance in this statistic, and we found that it was simply equal to the mean number of molecules counted, which was 4 pi a times c naught times dt. And so we can put these things together. And we find that this relative error comes out to the square root of c naught over 4 pi a cubed times g squared dt. Now, there was a bit of a debate, I guess I could say, about what this expression would look like before we calculated anything. In particular, we sort of did simple dimensional analysis and thought about what parameters this expression should depend on. And there was a little bit of a question on whether the C naught would be on the top or the bottom, right? Not if this is bringing back memories. Okay. So it's on the top. And that means that the larger the background concentration, the more the error will be in trying to determine the gradient. And can somebody remind me what the argument was, the prevailing argument was for that hypothesis? Why is that true? I know it's been like 14 hours or something, but a lot has happened in those 14 hours. Which were? There were two arguments.
One from dimensional analysis and one from just saying that sensing is going to be tough if the background is very high.
Yeah. Well, I don't think dimensional analysis can tell me whether C goes on the top or the bottom alone. I mean, because I could put C on the bottom and then throw in some factors of A. I mean, there are limits to what's physically reasonable, but apart from time, where we have only length here, so that was why it was ambiguous. But indeed, that's what I was going for. There was the argument that it's harder to measure a difference on top of a large background than a small background. And I think we can sharpen that a little bit to see whether or not that's actually going on here. So to do that, we can sort of simplify the situation even more, as was suggested yesterday, we can imagine that this cell really is, or we can ask, what if the cell really is just trying to count the number of molecules on one, detected on one half, and subtract that from the number of molecules detected on the other half. And just to do sort of like a simple intuitive calculation, we can go back to the thinking of Berg and Purcell with the perfect instrument and imagine that instead of an absorber, we have a monitor, right? So imagine molecules are now permeable, or the membrane is now permeable to the molecules. And the cell has some magic computation inside that literally just subtracts the number it detects here from the number it detects here. Okay? So now our estimate is not some asymmetry in the sort of angles of absorption, but rather we could say our estimate is just, if it counts N1 molecules in this half of its volume and N2 molecules in this half of its volume, because the concentration C2 is greater than C1, then what's the error it makes in doing the subtraction, right? So once again, we just need the mean and the variance here. The mean, so if I think about delta n, the mean is just delta c times the volume of either one of these halves, right? Now, I just want to sort of do an intuitive calculation, so I'm not going to worry about factors. The cell has a radius a, so the volume is roughly a cubed of either one of these halves, right? So this will be a cubed times delta c. So this is a mean. And the difference in concentration is, I can take like sort of a central point of this half and a central point of this half and ask how far they are apart, and knowing the gradient, that gives me the difference in the concentration, right? And once again, they're roughly a distance a apart, right? So this would be a cubed times g times a, right? So that's the mean difference that the cell is trying to estimate in this model. The standard deviation, or the variance, of delta n. Well, delta n is just n2 minus n1, right? So the variance of a sum or a difference of things is the sum of the variances of those things. So this would be sigma squared n1 plus sigma squared n2 plus any sort of cross correlation between them, right? That's just a result from statistics. And there will certainly be cross-correlation because these molecules, one that was here might end up over here, right? And I'm going to completely ignore that, nonetheless, okay? Because, I just want to know whether our intuition is correct, right? So I'm being fast and loose for this part. So I'm going to ignore any sort of covariance term, okay? Moreover, the number of molecules here and the number of molecules here are going to be roughly the same so long as the gradient is sufficiently shallow, and I would argue that's sort of what we're most interested in, the sort of detection limit. And unless we care about their difference, which we did for the mean, here we sort of just care about their sum. This is going to be roughly twice the variance in the molecule number as if we were counting at some intermediate spot. Okay? In other words, summing these two things, I'm not going to really care about exactly where we are in this cell. And moreover, I'm going to just reject that too, neglect that too, cancel it. So the point is that the variance, the error made in measuring the difference, is on the same order of the error made in trying to count either one of these halves, the molecules in either one of these halves. And that should be equal to simply n bar in either one of these halves divided by the number of times you get to measure it, right? T over this characteristic diffusion time, which is a squared over d. Right? And n bar is just C naught times A cubed. One of the A squareds gets popped upstairs, so this becomes an A to the fifth, sorry. A cubed times A squared is A to the fifth. And we have a dt on the bottom. Okay, so we have the variance, and we have the mean, and we can therefore compute the relative error. And we get, if I haven't done anything wrong, the same kind of scaling. So I hope this helps motivate why we get this in a sort of model-independent way, right? Here, I'm more literally suggesting that the cell is subtracting two quantities, and there's error in each of those quantities. And this is saying that the size of those quantities, if we increase the size of those quantities while keeping their difference the same, then the error in that subtraction is higher. A good context for this, another context that maybe is familiar to people, which was actually pointed out to me by Ilya back in the day, is if you write code, and I think all of you write code, certainly like probably last night you were writing code, then it's good practice if you ever find yourself in a situation where you need to find the difference between two numbers, that difference is going to have less numerical error if the numbers themselves are small, right? If you have like double precision integers or whatever, floating point numbers, and they're both really big, but their difference is like, 0.0005, yeah, that's not going to work, right? And so this is sort of a statement in that spirit, right? It's easier to find the difference between two noisy things if those things' magnitudes are closer to that difference, not farther.
Sorry, can you repeat that?
Yeah, so like sometimes you're writing code and it's bad code, and you end up trying to find the difference between two very large numbers, even though that difference is small. And you run into horrible numerical errors, and you have to think harder.
Oh, okay.
Right? This is like trying to find the small difference between two large numbers. If those large numbers have any sort of noise, which in the computer is just numerical precision, then it's going to be harder. But if those numbers themselves are close to that small difference, it's going to be more precise.
Those numbers themselves are close to that difference. So if you're saying the numbers are of the order of the...
So try to subtract 1,000,000,001 from 1,000,000,000,000 in a computer. It might go wrong because you don't have enough digits.
Okay.
But try to subtract 6 from 5 in a computer. The difference is still 1.
Got it.
It won't be as bad. That's my point. Does that make sense? Yeah.
What sort of numbers are we dealing with here?
Well,
How many circles would you expect from these circles?
yeah, so with amoeba, it's not so bad. I'll tell you as we go about a situation where I was looking at collaborators' data where the gradient was very small, and we were talking about like in this context, each of the two halves of the cells would correspond to volumes that contain thousands of molecules, and the difference would be only like 3. So it is important, right? It's a small difference on top of a large background for a lot of these cellular tasks. Okay. Questions about that? So we started this question or this topic based on frames from a movie that I drew about amoeba, right? And so, of course, the overarching question is whether or not, or to what extent this bound on the error is actually observed in the behavior of the amoeba, right? And I motivated for you these experiments where the amoeba is tracked at a certain distance from the source of the gradient, and it's the precision with which it moves is summarized by a statistic called the Chemotactic Index, which is simply the average of the cosine of the angle between the right direction and its chosen direction. The data show, as you might expect, that the farther it is from the pipette, the worse its chemotactic index is, until finally it gets to 0, which corresponds to absolutely no preference in migratory direction. So, so I've been sort of following this paper by Anderson-Wingreen, and one of the things they did in that paper is they translated this expression for the relative error in the estimate of the gradient to the chemotactic index, right? Because if depending on how well you can determine the direction you want to go, you're going to have some error in the angle that you make as you migrate, and that directly translates into the chemotactic index. So I won't write out the details, but they basically got an expression for the chemotactic index in terms of these parameters, and that allowed them to fit that expression to these data. The fit is decent, and the fit requires a fit parameter, and that fit parameter is basically a combination of some of the parameters in the problem, and given the fact that it was possible to estimate the gradient and the background concentration from the profile of the signal coming from the pipette, and the molecule was cyclic AMP, and the diffusion coefficient was roughly is well known for cyclic AMP, then from this fit parameter implies the only directly unknown thing, which is the integration time of the cell. So this fit parameter gave that the cell should be integrating for about 3 seconds. And the point I want to make here is that that's reasonable. That's not orders of magnitude off. I mean, the statement that applying this theory to these data is making is that if this is a bound that the amoeba care about, if they're coming close to this bound, then we should observe that they have to integrate their measurements for at least 3 seconds. Now, actually, it's important what Massimo said yesterday, right? Because all of our calculation assumes that it waits there and sits there for 3 seconds to acquire the information and then chooses to go, right? That's not what's happening. And so this bound is actually a little bit not fundamental, right? There's maybe a factor of 5 or 6, as he said, by which this is reduced. So maybe this should be more like, 1 second or a fraction of a second. But the point is that you can watch the movie and they respond they sort of change direction within seconds or tens of seconds. Other people have done more molecular experiments where you can sort of supply them with a signal and then use a fluorescent reporter to see when some protein involved in forming the pseudopod or whatever lights up. And that happens on the order of 5 to 10 seconds. So it's not like they are taking hours to respond. Right? So whatever internal machinery they have isn't adding that much noise on top of the fundamental noise that's simply present from detecting the environment in the first place. So, so loosely speaking, it appears that both in concentration sensing with my argument, not mine, but Berg and Purcell's argument with bacteria, and in gradient sensing with this argument for amoeba, cells appear to really actually care about and potentially have evolved to these fundamental limits. Okay. So, questions or comments about that? Because now I'm going to move on to talk about multicellular phenomena, and I'm going to start by telling you a little parable. And this parable comes from roughly 2007. And I'm calling it a parable because, I'm simply going to recount a result from a paper like I have been so far, but the papers I've been recounting so far have been mostly theory, and so I can call those lectures. But I'm a theorist, I'm not an experimentalist, so I'm going to attempt to describe some experimental results and probably, not get them all quite right, and so I'm just calling it a parable. So here's the parable. So the parable comes from work of Thomas Gregor, Bill Bialik, others involved in their work on fruit fly embryos since the time of 2007, probably before, but this is sort of the paper that I'm thinking of. By the way, Gordon was saying that he had the unfortunate challenge of describing Greg's work in front of him. Notice that Bill's not here anymore. And actually, tomorrow, if all goes right, my plan is to talk about David Schwab's work. And he's gone too. So. Okay, so the parable goes like the following, right? So many of you probably know, some of you probably even work on a developing fruit fly embryo where a lot of the action happens during the 14th nuclear cycle, where the cells are not full cells yet, they're cell nuclei, and they've undergone 14 divisions, and they're kind of arrayed in a nice way inside of this embryo, and they're beginning to differentially express different proteins that will ultimately define the fates of these cells and constitute the body plan of the organism. And one of the most well-studied expression phenomena is a pair of morphogen and downstream protein, which is bicoid and hunchback, right? So bicoid is a morphogen, which means that it varies over space, over the length of the embryo. It's a molecule that varies over space. And it does so roughly exponentially, okay, with a length scale of about 100 microns. Okay? And at a certain point in the embryo, right, so if we imagine sort of a line of cell nuclei within the embryo, cells express more of a protein called hunchback on one side and less of a protein called hunchback on the other side. And hunchback has binding sites for bicoid, and so sort of the textbook model is that bicoid activates hunchback, and so if you're above a certain threshold of bicoid, or if you're a cell that is measuring above a certain threshold of bicoid, you express hunchback. Below it, you don't, right? And that threshold corresponding to this boundary position is some number in nanomolar, but a nanomolar is about a micron, 1 over a micron cubed, so it's about, I'll call it C naught, and it's about 5 molecules per micron cubed. Okay. And the argument of theirs went like this. So these cells, well, these cell nuclei are about 10 microns wide. And so for a precise boundary to form along a line of these cells, then any given cell needs to detect the concentration it's detecting with a precision corresponding to the ratio of the cell size to the profile length scale, right? So the precision needs to be greater than, no, I'll call it, let's do in terms of error. That's how we've been doing it, right? The error needs to be less than A over capital lambda, which is 10%. Okay? And what's actually happening is that molecules of bicoid are binding to the binding site on the DNA, the promoter site of hunchback, and that essentially these authors thought of as a Berg Purcell problem. They thought of it as the same sort of problem we were thinking of with sensing a uniform concentration in the first lecture, right? So this error, which I'll call epsilon, should take the form of the error in estimating the concentration of bicoid, which should go like 1 over square root of S, which I'll define in a second, times C naught times D times T, right? And S is simply, right, so what I described is what's going on in one of these cells is there's some site on the DNA to which bicoid molecules have to bind, and that site is a couple nanometers big. So S is about 3 nanometers. Right. And the diffusion coefficient of bicoid in the embryo was also measured. It's rather slow inside the embryo as compared to within water, for example. It is about a micron squared per second. And if we call this, if we set this error to be 10%, and we know S, we know C naught, we know D, then we can solve for T, and we get T comes out to about 2 hours. And this was a problem, and the problem is because the embryo has really only existed for about 2 hours at this point. Moreover, as I told you, the nuclei have divided about 14 times. So the nuclei in their current positions have existed for much less than 2 hours. So these authors argued that something else must be going on in order to reduce the error beyond what's possible here, because this is supposed to be a fundamental bound. That's been the premise of these lectures all the way up until now. So how is this system beating this fundamental bound by so much? That was the question that they asked themselves. Any ideas on what answer they proposed? Yes? Something is sequestering them to the site where they need to bind? Or no? To reduce the free concentration. Like the effective C naught might be lower than, well, I would say that might work if it made it higher. Maybe that's what, right? Because, sorry, we don't want to get, I'm jumping back and forth here, but a higher background hurts if you're trying to sense a gradient. Even though this is a graded profile, the baseline assumption is that the cells are trying to sense a concentration. Yeah, it gets a little, that took me years to figure out, by the way. So it's like, this is a graded profile, but we're imagining each cell has a task, and that's sense my local concentration. Yeah. So sequestering might help if it reduces the local increases the local concentration. Other ideas? Kill diffusion?
Replace it with heat energy. Replace it with an active process. Then all the bounds go out the window.
Kill diffusion. You don't mean reduce diffusion. You mean using a different mechanism. Yeah.
Heat energy and then no bounds of that.
Yeah, actually, this is something that one of my students and I have been thinking about a bit, and that is that, right, actually, in this case, there's pretty good evidence that diffusion of bicoid is what shapes the profile ultimately. But indeed, if there are other active processes like, some sort of tracks or some sort of, ballistic movement mechanism for bicoid molecules to get from the source end of the embryo to the place where they end up, then that would invalidate this physics, right? So that might decrease the error as well. And in some systems, not this system, but in some systems there are sort of active processes. The cells can make protrusions, long-range protrusions to other cells in the developing tissue. Yes?
So I've always wondered, and I don't know the answer here. Maybe if we just spend energy on producing Bitcoin on one end and then spend energy on eating it on the other end, right? So, and then it diffuses freely in the middle, but in some sense it diffuses under chemical potential, right? So it eats energy out of chemical potential. Would that help?
Yeah, so just to fill in what Ilya is asking a little bit, so what creates this underlying exponential profile, the assumption is that Bitcoin is produced at a constant rate at one spot, it diffuses, isotropically, and then it has a uniform chance of degrading anywhere, right? And you set up that problem, you get an exponential profile in one dimension. Ilya is suggesting instead maybe it isn't degrading anywhere, it's all degraded at the end, if I understand correctly. Or for example, some sort of non-uniform degradation. So in that case there'd be kind of, well, in this case too, but you'd be modifying the flux from left to right.
And so the flux is not anymore the Berg-Purcell flux from stationary constant concentration. Maybe one can cook up a gradient profile where that will have an effect. I don't know.
So here we have diffusion everywhere and degradation everywhere, and it is still, the number of molecules in any given volume is still Poisson distributed. There, you'd be saying, well, take degradation out, so you just have diffusion everywhere, except at one end of the embryo you have a lot of degradation. I still think it'd be Poisson, right? Yeah. But okay, so maybe there's other sort of active things you could do to modify the Poissonian nature of the number of molecules counted. Other ideas? That was one of their proposals, that the cells would be talking, right? So this assumes that it's a local computation that each cell is doing. All it's doing is it's ignoring everything else, it's just trying to read something out. If it got some information and then the cell next to it also got some information and they had some way to share it, then that could potentially be an error reduction mechanism. Because if you average a bunch of noisy numbers, you get a less noisy number. And so they propose that. I don't think the story is clear. Like I said, this is a parable, so if you know that there's been more knowledge in this system, then please raise your hand. But yeah, so one of the things it could be is the molecule hunchback itself, right? So this gene expresses hunchback proteins. And it could be that not only sets the fate of the cell, but also hunchback is diffusing itself and sort of being shared among other cells. And you can sort of think about this for a minute, and you can say, yeah, that will help because averaging reduces the noise. You have to be a little bit careful, though, because if all the hunchback produced by every cell was shared, almost instantly and infinitely with all other cells, then you wouldn't have this boundary pattern, right? Then actually you would defeat the purpose of reading out a graded profile. So there's a balance there, actually, and this has been thought about theoretically in the intervening years, and indeed, there is a balance. There's an optimal hunchback diffusion coefficient that's pretty reasonable and does reduce this, would reduce this by a considerable amount. So that's one of the things they proposed, and that's sort of that. I want to use that to motivate a more general question, which is, can we actually calculate that? If cells start to share information with each other, then how does that affect the precision with which any given cell knows the statistics of its environment? So I want to start to broaden our discussion to multicellular sensing. Okay, so the general question is, how does cell-cell communication modify the sensory limits that we've derived so far? So, I want to start simple. I want to imagine now, any sort of generic system with a bunch of cells, maybe at particular positions, and they have the simpler task that we've been discussing, which is they want to detect a uniform concentration. call it C naught. And we want to know the precision with which they do that. But now, let's imagine that they have some way of talking. And we can indeed stick with that simplest way of talking, which is just to have some molecule that they produce be diffusing with the potential for it to be read out by other cells, right? So imagine now each cell is also secreting molecules that can be read out by other cells, or maybe even the same cell. That's not really a different color. Yeah, good call. So this is some sort of communicator molecule. To help with sensing the signal. Okay, so we're going to calculate something, but as always, we should think about what we should expect first, right? So with one cell, we had an error, which was 1 over the square root of A C naught dt. This is with one cell. Now I'm going to assume we have, say, n cells. And, I'm not going to, we can think about, where they are and, how much they secrete and stuff like that. But let's just start with, some hypotheses about how this error might depend on n. So I think it would be reasonable to expect that it should not go up with n, right? Square root, maybe. So why do you say square root? It's counting. Yes, exactly. Or that is, well, it's a counting problem in general, but yeah. Right, when you have n independent measurements and you're doing some sort of averaging, and we're assuming these cells are averaging their information, then it's like a counting problem, and it goes down like 1 over the square root of n. So maybe this goes like 1 over n a c naught dt, absorbing the n into the square root. Okay. So good. That's a reasonable hypothesis. So with that hypothesis in mind, let's actually consider a specific scenario. Now, there are many ways that cells communicate. And for our purposes, I'm going to sort of lump them into two broad categories. One is, as I've drawn here, what I'm going to call long-range communication, right? Cells are not touching each other. They secrete something into the environment. It diffuses around. It can be taken up by other cells. So that happens all the time with many single-celled organisms. Bacteria do this to perform a function called quorum sensing, where the signal is sort of conveying information about the local density of other cells. Amoeba do this, right? The experiments I was talking about where cyclic AMP was the attractant, that is a molecule that's secreted by other amoeba. So they secrete it and then they're attracted to each other and they aggregate. But the other type of broad category of cell-cell communication is short-range communication. And that might have been more appropriate to describe, a line of relatively close cell nuclei that is presumed to sort of just be communicating among neighbors. So I want to start with short-range communication. And there are, as you can imagine, plenty of other examples, one of which I'll talk about today if I get to it, of short-range communication that do not rely on, passive diffusion among still-forming cell nuclei. For example, cells have structures, like in tissues, when cells are next to each other, they have structures called gap junctions, which are basically like channels that molecules can pass from membrane to membrane. Yeah?
This picture of the multicellular
Yeah.
molecule, is it because they have just a higher concentration of the communicator molecule, or do you have multiple different types of communicator molecules and you're doing some basis on that? I see many of these different types of communicator molecules and doing sensing on that? Or?
Yeah, so that's a really great question, and that's something we're going to have to think about, which is like how complex of a communication scheme do you need? And the answer will usually depend on what the task is. And so here the task is to sense one number, which is the background concentration. And so it will suffice to just have one communicator, right? But as we'll talk about, we'll talk also about multicellular gradient sensing, where the task is to sense sort of a direction and a magnitude, right? So the loose way of thinking about it is that's two numbers. And so it will turn out that that's not possible to do with one communicator. You sort of need two variables to do that, right? So, yeah. 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 Yeah, that will become explicit, and that's important, right? It needs to convey the information it's receiving, and so it needs to depend on the sensed concentration. I'm not going to assume any, but that's a very interesting open question, I would say. As soon as you introduce regulation like that, then you have a nonlinear system, and if you haven't realized yet, everything I've done and will do is linear to exploit Fourier space, and everything is basically Gaussian, and just makes life easy. Okay, so I want to start with short-range communication, and I want to consider a simple case first, and then I'll sort of just tell you the results when we generalize. And that simple case is just two cells. And the model I'm going to set up is the following. So the cells are, they have radius A, right? And they're touching, which means that they're 2A, their center-to-center distance is 2A. I'm actually going to go a little bit beyond the perfect instrument and the perfect sink and actually endow these cells with receptors that we're going to model. And these receptors are sensing the background concentration. So basically a molecule can bind to a receptor, sit there for a little while, and then unbind. So I need to give rates for these processes. So I'm going to call the binding rate alpha and the unbinding rate mu. And of course these, the concentration, is a fluctuating thing. I'll call it C of x and t. But on average, in the absence of these cells, or even in the presence of these cells, because binding is an equilibrium process, the mean steady state concentration is this thing C naught. And molecules are diffusing with coefficient D, of course. And then we have to account for what was just asked about, which is what's the communicator and how is it related to the sensed signal. So the answer in this model is that if a receptor is bound, then it has a rate of producing an internal molecule, and that rate is beta, and that internal molecule can also degrade into nothing at a rate nu. And that internal molecule is indeed, it's the internal readout of the external sensed signal, and it's also the thing that's going to be communicated. Okay? Which means I need to allow it to be exchanged between these two cells. And the exchange rate I'm going to call gamma. Okay. Is that clear? So I have C for my external concentration variable. I'm going to say this is cell 1 and this is cell 2. And the number of bound receptors on either cell 1 or cell 2 I'm going to call R. So R is a sort of a fluctuating function of T. And the number of communicator molecules in each cell I'm going to call M for messenger molecule, for example. Yes?
So why is the number of receptors changing?
It's fluctuating. So in principle, we are only going to care about steady state, but I'm going to write down the equations right now, and there's going to be noise terms. And so strictly speaking, these things are time dependent due to fluctuations, and that's sort of necessary to do the Fourier analysis, right? The spectrum has a frequency.
But is that what's happening in the cell? Like the receptors are removed and then you get...
Oh, no. This is the number of bound receptors. Ah. Yes. So it's only fluctuating because the number of molecules is fluctuating. Yeah. Sorry. This is the number of bound receptors. Yeah. Yeah, in principle, there's a total number of receptors, but I'm going to immediately assume that that total number is larger than the number of bound receptors so that I don't have to worry about saturation effects. And it is fixed, but it doesn't matter because it's big and I'm going to neglect it. Yeah. Okay. So I'm going to take the same approach for this model as I did for the perfect instrument, and that is to write down Langevin equations. And I won't take you through the details. I'll just sort of give you the result of finding the power spectrum, taking the low frequency limit to get the variance in the long time average. And in this case, we'll be interested in the long time average of M. M is the cell's internal readout that contains also information from the other cell. Yes?
I was just curious, like, which of these parameters can be measured experimentally? Because I was just concerned, like, there are a lot of parameters here.
Yeah.
Maybe this theory is just inherently not falsifiable.
Yeah, great concern. Great concern. And in fact, I will show you, I won't show you, I will draw for you data that we used when we talk about gradient sensing, that we used from an experimental group to compare our model to. And that still might not actually answer your worries about whether this theory is falsifiable. But to answer your more direct question, I'm going to assume that since you haven't asked it so far, that you're okay with the idea that background concentration, diffusion coefficient, these external parameters, and certainly cell size are fine. I mean, people can measure those. The next question is, what about binding and unbinding rates and the numbers of things inside of cells? Well, frankly, people can measure those, and there's a lot of, I mean, that's the subject of a lot of experimental work, I would say. But for the system that I'll describe specifically either today or tomorrow, we only have estimates, right? And so one of the things you can do, and that's sort of the approach I'll take, is that we will see that each of these sort of steps of the model will contribute its own lack of precision, its own errors, right? And so indeed, depending on the values of these binding and unbinding rates and the production and degradation rates of things that maybe we aren't able to measure, they will contribute noise on top of the things we've been considering so far, which is sort of like the molecule counting noise. So at the very least, you can say, you can ask, if the cell has a ton of receptors and it really has a ton of internal molecules, is there still a limit? And how does that limit scale with the number of cells? That's what I'll be focusing on. Other questions? Okay. So the Langevin style model of this process is the following, right? So we have a diffusion equation to describe the external signal. But now the receptors act as sources, as sinks and sources of these molecules, right? So that modifies the concentration. And so I'm going to do something offensive again, and that is to treat in this equation each of these cells as a point particle. Okay? Okay. Hold your objections for just a second. So that is to say that at a position x1, there will be binding and unbinding rates that act as sinks and sources of this concentration. And so that will be given by the dynamics of the bound receptors at cell 1. And then, of course, we have the same thing at cell 2. with R2 dot. And then, just as before, there is a Langevin noise term to account for the particulate nature of the molecules that are diffusing. Okay. The thing I wanted to say to maybe reassure you is that, so this is bad, and it's particularly bad because it ignores a parameter that's going to be fundamentally important in our result, and that is A, right? These cells have a size. They need to count. A will come back, okay? You're not going to see an A in this model, but I'll describe to you how it will come back. Okay. So I need to tell you what the dynamics of the receptors are. So the dynamics of the receptors, so R1, for example, is alpha times the concentration at position 1 and time t minus mu times R1, right? And it also has its own noise term, which I called eta 1. And of course, this is the same for cell 2 as well. Now, here's where I'm neglecting receptor saturation, right? Because in principle, the rate of binding receptors depends on the number of free receptors, which would be like capital R minus R, but I'm neglecting that. That's all being shoved into this alpha. Okay, and then finally, bound receptors can produce internal messenger molecules. So I have M12 dot is beta R12 minus nu M12 from the degradation, and then I need to account for the fact that molecules can be exchanged, right? So I have an exchange rate gamma. Molecules can come in from the other cell. They leave from that same cell. And there's also a noise for these reactions as well, which I called chi. Okay? All right. So just to be clear, we can't solve this unless we specify the statistics of these noise terms. They're all zero mean, as we have been working with. The statistics of this eta are exactly the same as we saw before. Namely, it's a strength of 2 C naught D. It's delta correlated in time. And it's not delta correlated in space. It has these gradient operators that account for the correlations between neighboring locations as molecules diffuse. Okay. These are a little bit easier. The statistics of Xi, which is just time dependent, well, time and cell dependent, are 2 mu r bar times a delta function in time, right? When these binding and unbinding reactions fire is not dependent on the previous one, so long as the receptors are considered independent, and delta correlated in terms of cell identity. And if you're curious about the strength of this noise, it comes from the fact that there are 2 reactions in this process. There's a binding reaction and an unbinding reaction. That's where the 2 comes from. And the propensities of both, the propensity of the unbinding is certainly mu r on average, but so is the binding because this thing is in steady state. In steady state, on average, these 2 things are equal to each other. So that's the 2 mu r bar. R bar is alpha C naught over mu. Okay. And then finally, we have this chi, and chi looks very similar to Xi with one exception. So it has this 2 m bar delta T minus T prime times the nu. So it has the nu m bar propensity from the degradation and also from the production. But of course, we also need noise terms from the exchange reactions as well. So that actually has a gamma m from here and a gamma m from here. So we get a plus gamma here, delta ij. I'm sort of going through this in detail because, I mean, it's sort of it's not very intellectually stimulating, but there's a systematic way to set these things up, and if you don't do it right, you get the physics wrong. So, and there's one more term here, and that term is important. That term is needs to account for the fact that when a molecule leaves cell 1, and there's a noise associated with the propensity for cell 1 of that happening, it also enters cell 2. And so that reaction should induce correlated noise between cell 1 and cell 2, which means that this Langevin noise term needs to not just have a delta term. It needs to have a term that connects cell 1 and cell 2. So there needs to be some term that is proportional to 1 minus delta ij for the other cell in this case. It's going to have strength gamma, and it's going to be negative because of the anti-correlation I described. And all of this is hitting the m bar and the delta correlation in time, right? So I hope it's not too hard to imagine, because the origin of this, or the physics behind these anti-correlations, is the same as the origin of these sort of gradients in space. In other words, you could imagine this is two sites on a diffusion process. It just happens to be diffusion between cells. But if we then tried to make a lattice for the diffusion of the external molecule, it would be like a whole lattice of such processes. And you'd have a lot of these anti-correlation terms, and they would become these in the continuum limit. So this is just to give a flavor of how you get all these things right. Yes?
Is it possible you can just like walk through the model again, just describe
Sure.
where each term is coming from?
Sure. And just because I didn't mention it, m bar is simply beta r bar over nu. Okay, so this is a model for the diffusion of the external chemical that's uniform on average, but it gets sunk and sourced by the receptors, right? So if you have a receptor binding to that molecule, that should decrease the amount of concentration, and so that's taken care of here. Okay? Any receptor dynamics is going to have the inverse effect on the concentration.
And the delta?
The delta means that it happens only at site 1, and site 1 is sort of the collapsed version of cell 1. Okay? Same thing for cell 2, noise. Receptor binding.
So C is a function of only x1 and x2.
C is a function of all x space. C is a field, right? C is a field in space and time with noise.
Okay, so C is outside, like
Yes.
the ether.
Yes, absolutely.
Okay, and then there are only x1 and x2 locations within all of that
Yes.
space.
X1 and
that
X2 are
representing the
constants.
cells.
That's right.
Okay.
So yeah, I should have been a little more explicit. So this is point x1, this is point x2, and the only thing we're going to really need to know is that the distance between x1 and x2 is a diameter, because the cells are touching. Okay. And then this is the binding propensity. It happens at a certain rate, and it's dependent on how many on the molecule concentration near that receptor or near that cell, more specifically. And then you unbind based on the number of receptors that are bound. Noise. Then bound receptors can produce messenger molecules at a rate beta, and those messenger molecules can degrade, and those messenger molecules can be exchanged among the two cells, between the two cells. And there's reaction noise in those processes as well. Other questions? Okay.
These noises don't talk to each other.
These noises don't talk to each other. That's correct. Yeah, they don't talk to each other except chi. Chi in cell 1 talks to chi in cell 2 because a reaction changes m in both at the same time.
What I'm saying is that the
Yeah, there's no reason to do otherwise. Yeah. Okay. So, like I said, I'm going to spare you the details, but we go to Fourier space, we find the power spectrum in particular of M, and the thing we want is essentially, yeah, let me go to the next board. So the thing we want, this thing which I've started to call epsilon for error, and I'll square it, is the cell's estimate of that background concentration, which we're going to now take to be the variance in the long time average of M over the mean in the long time average of M. And this we get from the power spectrum of M in the zero frequency limit divided by the averaging time, so long as the averaging time is much longer than any of the intrinsic timescales. And at the end of the day, you get the following result. I'm going to write it in terms of an important ratio, and that ratio is the ratio of the exchange rate gamma to the degradation rate nu. And the result is a term for corresponding to this molecule counting noise that we've been familiar with this whole time, because it has the scaling that warms our very hearts. And another term that does not depend on those things, but rather depends on parameters of the binding and unbinding process, specifically mu and r bar. And finally, a term that depends on u and parameters of the messenger production process, namely nu and m bar. Okay. Okay. So we got three terms out of this model. That should make sense to you because there are sort of three stages. You have to count the molecules, you have to have a certain number of bound receptors, and then you have to produce messenger molecules. This term I think we can understand. All three of these terms depend on this ratio, which I would characterize as the strength of the communication in the following sense. If cells maximally share their information, gamma should go to infinity and the relevant other timescale is nu. So this ratio should go to infinity. And then we have these other terms, and so most of the time people call this the extrinsic noise. It's the noise that comes from molecule counting that's sort of unavoidable because it's part of the environment. And then these are associated or referred to as intrinsic noise. And they come from, in principle, something that the cell can control. And this relates to your question, how falsifiable is this? What parameters can you measure? So there are additional sources of noise, and they come from the fact that there are a finite number of receptors in the system, on average, in the cell, and a finite number of messenger molecules in the cell. But in principle, if either of those were ramped up by the cell, that would diminish these sources of noise. And in fact, that's what I mean when I say we're going to focus on the extrinsic term from here on. And by the way, there's a nice interpretation of this, right? So as you know, receptors are being bound and unbound. Messenger molecules are being created and degraded. These are both birth-death processes, at least if you ignore receptor saturation. And birth-death processes are Poisson processes, so you would expect that their relative error, the variance over the mean squared should go like 1 over the mean. Okay, that's the origin of this r bar and this m bar, except we're doing a time average, so it's not just 1 over the mean. The variance is also reduced by the number of independent measurements that you can make. Well, how many independent measurements can be made in the context of the receptors, well, it's the total averaging time divided by the intrinsic time scale of the receptor binding and unbinding process, which is the unbinding rate. And similarly, it's the total time scale divided by the degradation rate for the messenger molecules. So this is like Poisson noise time averaged in both of these cases. Okay. So, but the important thing is that the communication affects everything. All right? So the communication affects everything, and in particular, we're going to focus on how it affects the extrinsic noise in order to see whether we were right about this scaling. Okay? I mean, n is just 2 here, but. So yeah, let's see if we were right? So let's think about some limiting cases, right? So this expression, just the extrinsic noise, looks like 1 over 2 pi a c naught dt times a factor that's dependent on u. But let's think about limits. Well, if u is 0, that corresponds to the cells not communicating at all. Okay? So that should sort of reduce to basically what we've been looking at previously, except my model's a little different, so maybe it'll change a prefactor here and there. But if u is 0, you can see that this reduces to 1. Okay? Which means in this model, if you care about prefactors, the prefactor is 1 over 2 pi. That's in contrast to the perfect absorber, which is 1 over 4 pi, or the perfect monitor, which is 3 over 5 pi. You know, I mean, who cares, right? What's interesting is how this changes, or sort of the relative effect of communication. So let's make communication in contrast as large as possible. In the limit of infinite u, then you just have these squared terms, and we get 3/4. Now, is this consistent with what we thought? If I change this to epsilon squared, and I plug in n equals 1 or n equals 2, do I get the right ratio? No. No. If a single cell can get a prefactor of 1 under this definition, and then we have 2 cells perfectly sharing their information, I would have thought we would go down to 1/2. So the question is, why did we not go down to 1/2?
They're probably sensing the same stuff.
Yes, they probably are. They're right next to each other, right? And the thing they're sensing is mobile. And this model captures that, you know. And so some of the molecules detected by cell 1 indeed go on and bind cell 2. These are not independent measurements. So we can't get that full reduction we would expect for independent measurements. We only go down to 3/4. And in fact, if you extend this to more than 2 cells, well, first of all, you have to decide like how the cells are arranged. And so, you know, maybe the most natural thing or the simplest thing to do would be to arrange them in a sphere as best you can, like on a lattice, for example. So for a larger n, what you see if you plot, say, the log of epsilon squared versus the log of n, right? So here was one, okay, no, I can't label it like that. But anyway, here was a single cell, you know, 2 cells that went down to 3/4, not 1/2. If it had gone down by 1 over n, we'd have something like this, 1 over n scaling. But for this system, we see something like this. And the scaling is 1 over n to the 1/3. Right? So this issue of counting adjacent portions of the environment, and I've assumed, by the way, that these cells are like not blocking the signal from other cells. I mean, there's issues there, but that I'm ignoring. But, you know, just the fact that their measurements are correlated with each other prevents you from getting what you might expect from independent measurements. And it goes down much less steeply. Yeah?
Okay, so here I think that distance, I mean, these things were touching each other.
Yes.
Okay, so what if I just separate them by a reasonable distance? So
Yeah.
they're not sensing the same
Yeah.
thing? Would
Yeah.
I expect them to get to get this counting? Like,
Yeah, so remember I told you that there were two types of communication we're going to consider? So that's absolutely why we should next consider long-range communication, right? Let's separate them out so that they sample less correlated parts of the environment. There's going to be a trade-off, though, right?
of course,
Because
the farther they
yeah,
are, the more difficult.
exactly. So that's the trade-off we want to explore. Before we do that, can we just understand this 1 over n to the 1/3? So I'm going to argue that we can, and we can do that in the following way. Let's imagine that this clump of cells is big enough for me to start to approximate it as one continuous sensor. Then the volume of this sensor is n, scales as Na cubed, which means the length scale of this sensor, which I'll call capital A, scales as n to the 1/3 A, right? Which means if we just, prescribed a Berg-Purcell expression for that, we would get 1 over capital A C naught dt instead of little a, and that's just the n to the 1/3. So in a way, it had to be this way, right? It had to be this way. But that is exactly the right question. So what happens if we allow cells to start to spread out? Then it's good because we're reducing correlations. It's bad because they might not be able to talk to each other infinitely strongly like we've assumed. Any guesses on whether it will be a better or worse drop-off of the error? 1 over square root of n, so 1 over n to the 1/2. So here, for long range, maybe 1 over n to the 1/2 is a guess. So you think it'll do better?
If it's not too far.
Yeah, if it's not too far. Any other? I don't know. Yeah.
Why is the fact that they're close to each other affecting their communication? I mean, they can send so many molecules out. I mean, there is no limits on that, right? So they can basically have the communication between them be effectively deterministic.
We have essentially assumed that communication is deterministic. It's not adding anything to the noise.
So why is
And now you're asking, sorry?
why does this affect things then? Right?
Well, because even perfect communicators are sampling correlated pieces in the environment. So it's not, the two measurements that they make and are perfectly sharing are not uncorrelated. Yeah?
When you're moving them far apart, why does the communication become a problem?
Yeah. Good question. So, right. So we need to talk about that. So, how do cells communicate when they're far apart? Well, the most common answer is that they secrete molecules that themselves diffuse in the environment, right? And Ilya's question is, a cell could secrete like a bajillion molecules, and then it could still be perfect communication. And no matter how far its partner cell is away, if it's not receiving communication strongly enough, well, just ramp up the secretion rate. We're interested in fundamental limits here. So what's the problem? So let's talk about that. Let's talk about long-range communication and start with that question. Start with that planted question. Because I assume you know the answer to this. Maybe you don't. Okay. So now let's imagine we have two cells, and basically let's keep things the same as much as we can. So they're still trying to sense something. A bounded receptor can create a messenger molecule. But now these messenger molecules sort of can be secreted, or the most simple thing we can do is just sort of make these cells permeable to them, so we don't have to worry about that. Now, let's say they just sort of diffuse out into the environment, right? They're still produced at a rate beta, In principle, they degrade at a rate nu, but in light of Ilya's question, we can even assume that they never degrade, right? That might be the best way to communicate. You want to flood the system with what you know.
Flood it, then you lose all time resolution, right? Because
Yeah, so that is a problem, right? The inherent in everything I've calculated is that the averaging time is bigger than any intrinsic time scale. And so if you set this intrinsic time scale to infinity, we have a problem. So instead, maybe, okay, nu is finite, but let's say you turn beta up to infinity. Okay. So the first question is, there's going to be some distribution. Let's just consider this cell only. There's going to be some distribution or some concentration profile for the messenger molecule. And I'll call that continuous profile rho, right? Rho of x. And now these things are diffusing. I need a new diffusion coefficient. I'll call it d prime, for example. So, just as a function of, let's say, this distance between the two cells, x, the question is, if you have diffusion, secretion from a single source, how does the concentration profile vary? Now, I would argue, and we sort of intuited, that it should go down as you get farther from the cell. And you can solve the diffusion equation with a single source in 3 dimensions, and you can get the expression. And the expression is, it should go like e to the minus, distance that you are from the source over some characteristic length scale, which I'll call lambda. And that lambda is simply square root of d prime over nu, right? Should get bigger with diffusion and smaller with the degradation rate. But in 3 dimensions, it also has a 1 over r term. So it goes like x minus x1, distance you are from the source, also on the bottom. And let me just get the prefactor right. So yes. So this will be rho as a function of x. And the prefactor is proportional to beta, because the more you secrete, the higher this is. More specifically, beta r bar, if these are coming from only bound receptors. And then we have a 4 pi and another d prime. Diffusion should also sort of dilute the strength of this signal. Okay, so now to return to your question, Ilya, I think you're basically saying, look, if you ramp up beta, can you essentially, make this thing become uniform, I think is what you're asking, right? You want to sort of send as much stuff out into the world as you can so that anybody, no matter how far away they are, is reading it, right? And the answer given by this expression is that you can't, right? So in other words, yes, you can ramp up beta. That increases the maximum of this curve, but it does not change the fact that it is a decreasing curve. And more specifically, another strategy you could do is say, well, just make your communicator messengers, your messenger molecules diffuse as quickly as possible. I mean, I don't know if that's a strategy, but that's sort of a limit you can think of in your head. And indeed, that does change the extrinsic length scale, and it sends it to infinity, but there's still this 1 over r fall off, right? So that's sort of the mathematical answer to your question. The intuitive answer to your question relates to something you said a couple days ago, which is that diffusion is sort of an inherently 2-dimensional process, right? And we were thinking about molecules diffusing into a 3-dimensional target. You can think of them as 2-dimensional membranes hitting a 3-dimensional target. Well, if diffusion is a 2-dimensional process, and we're asking it to fill 3-dimensional space from a single source, it's not going to work. And this 1 over r is a manifestation of that not working. So this is a surprising result unless sort of you've already, come to terms with it already. And that is that you can have a source in 3 dimensions that's continuously secreting things at as high a rate as possible, and you will never fill all of space, and therefore you can have a steady state. So there's no degradation. The degradation is just dilution into a space that it cannot fill, dimensionality-wise. Yes? Which relation?
The distance, basically the two cells are touching.
Oh, yeah. I should have mentioned that because I hinted at it only. When I went to Fourier space, notice there's no A in this on this whole board, right, or in this whole model. When I go to Fourier space, I turn from x and t to k and omega, and then I have integrals over k. And what happens is those integrals diverge unless I set a maximum value of k. That's a maximum value of the wave number corresponding to a minimum wavelength,
Which is 1/2,
which is A, or on the order of A. You can get the factor right if you just think of like, a simple limiting case or something. But that's how I reintroduce A into the problem as a cutoff in one of the k-space integrals. Does that help? Okay.
But inherently, like, there is no, like, these equations, like, I mean, presumably, like, in this
Yeah, no, but I think I understand your question better. I never explicitly use this result. So I could imagine that this model describes two cells that are an arbitrary distance apart, but they teleport their molecules from one to the other, which is not what's happening, and I would never want to imagine that. But the answer would be the same. That's my only point. However, no, I do use this result. I'm wrong. I do use this result because it's up there in the first line, right? I have a delta function of x1 and x2, and that's influencing a spatiotemporal field. And so x1 and x2 need to be located at a certain separation for me to solve this. And I do, and it's there. So A comes in both in this way and in cutting off the integrals which affect the volume of available to count.
But, like, I mean, the exact, the C dot here equation does not explicitly, like, rely on x1 and x2 being a certain distance from each other.
Not explicitly, but in order to calculate this, I need to at some point decide.
Right.
Yeah.
And so basically, I mean, what I'm trying to get at is that eventually you have to utilize this.
Yes.
Right?
Yes, exactly. So here I have a new variable, and that's L. Okay? And with two cells, or with more than two cells, it's a bunch of separation distances that are in principle arbitrary and that I could, set or optimize or whatever. And so what happens, I'll just give you the result, right? Because we're kind of waiting for it. Is it better or worse to do long-range communication? So to do this, I just need to tell you what's the difference in the equations I'm solving. So everything's the same with C and R. It's this M that's different, right? So the M equation gets now replaced with a field equation, which looks like a diffusion equation for the M molecules, their concentration being rho. The degradation term is still there. But now they get secreted at locations x1 and x2, and with strengths beta R1 and beta R2. And now there's noise associated with the secretion reaction as well. Oh, I've already used eta. Zeta 1, zeta 2, okay? And then there's noise associated with the diffusion and the degradation reaction as well. I'm running out of Greek letters, but I don't think I've used phi. Right, so this phi will have statistics very similar to eta does over there, except it'll have an additional reaction-only term involving nu, which will be delta correlated in space and time. These zetas will be delta correlated in time and have strength according to beta R bar, right? So just crossing the T's and dotting the I's. And then, you have a similar thing here, plus same for x2. In the rho expression, it's sort of the field from both of these cells, right? And you Fourier transform, and you come back, and there's one more thing. We need to define what's being counted. So we do need to define an M for each cell. And that's simply the integral over the volume of that cell, right? So it matters where it is, of rho. And I guess in principle, due to fluctuations, this is also a function of t. Right? So that allows us to, I mean, it gets pretty hairy with, you know, all your integrals, but that doesn't matter. This is a linear system. You can do it if you have enough resolve. And what we want is the same thing. We want the power spectrum of M, and what we find for once again, just the extrinsic part is we find that the relative error goes like, for two cells, 16 plus 9 L over A squared divided by 2 plus 3 L over A quantity squared times our favorite Bergen per cell term with the model-specific prefactor. Okay, that's just the extrinsic part of the noise. Okay, so as you predicted, Ahmed, this is a function of L over A, which looks like this. Okay, so there is some minimum, there is some value of L at which the noise is minimized, right? So this looks like either 1 if L is much, much greater than A. So this is sort of also answering Ilya's question. If you do take the cells infinitely apart, they can't see each other anymore. We get the 1. But it's equal to 4/5 if L equals L star. And L star is 8/3 of A. 8/3 of A is a little more than a diameter, right? So these cells are like hovering near each other. And they've sort of chosen to do that, or that is, it's optimal for them to do that because of this trade-off. They're now measuring separate portions of the environment to the extent that they can, but they need to stay close enough to communicate that information. And notice, we also don't get down to 1/2. In fact, we didn't even do as good. I mean, you know, just two cells. This is 0.8, that's 0.75. Okay, it's like roughly the same. The important question is what happens when you have more than two cells. And now you have a bunch of separations L, right? And you can put them anywhere you want, but you can also sort of ask the optimization question, you know, where should these cells be to collectively minimize the error in any one of their estimations? And when you do that, you find a curve like this, okay? And it's actually 1 over n to the 2/3. So they're exploiting this trade-off to do better in the long run than the short-range communication, but they still can never get to, you know, as if they were independent. They still have to stay near enough to each other to share. Yeah.
How do you deal with crosstalk between cells? So like you're effectively getting the signal of another cell because you no longer have a pair. So
Yeah.
your communication channel is not one-to-one. You're actually communicating with everyone. And so far we were only using one thing to do that communication.
So that's a really important question. How do you deal with crosstalk between cells? I think the reason why that's not a huge issue here, once again, is because you're trying to just estimate one number. And notice I've kind of swept a little bit under the rug here. All I'm ever caring about is the ratio of the noise to the mean. But I'm not really worried. That's precision. That's not accuracy. Accuracy would be whether I can interpret the mean for a specific concentration and have like a lookup table or something inside of my network or something. But basically, you can. I guess I can frame your question just with two cells. You can vary the distance between them, and that will change the relative error as we've been calculating. But it will also, frankly, it will change the mean. Because this cell will not just have its own molecules that it's producing, it will have more or less of the other cell's molecules. So that's a form of conflation maybe that is sort of a primitive version of crosstalk. So I think it's a problem already here, but certainly if the task becomes more complicated, then separating what is yours and what is information coming from some other cell is delicate, and we haven't accounted for what kind of networks do that here. Yeah?
Sorry, can you go again through the density equation that you were trying? Just to explain the delta terms?
I can. I think it's a good idea to say, thanks, that's it for today.